right, I'm going to explain to you the citric acid cycle, which is also known as the Krebs cycle. So let's go ahead and write down the name of the cycle at the top, citric acid cycle. And do remember that this is now part of the anaerobic respiration process. So if you can remember, glycolysis ended with us producing 2 molecules of pyruvate. Right? When we take 1 molecule of glucose and we break it down via glycolysis, it produces 2 pyruvate molecules. So pyruvate is going to be the first intermediate in the citric acid cycle. It is going to go into this cycle and take part in a whole series of events. Now, you can maybe write down here for yourself, pyruvate comes from glycolysis to help you remember. And there's a few things that need to happen for pyruvate to be changed into a molecule of Acetyl Coa. So you see, this is a coenzyme that attached here. So first thing that needs to happen is that coenzyme needs to come in. So let's write it over there. Coenzyme A is going to come in and it's going to attach to that pyruvate molecule, and then we're going to see a molecule of carbon dioxide leaving. Also needed is some reducing power. So let's put an arrow on that side and let's write this in. We're going to have 1 molecule of NAD plus come in. It's going to pick up a hydrogen from that pyruvate molecule and form NADH. All right, there is an enzyme here that is required to make this reaction happen. That enzyme is called pyruvate dehydrogenase. Pyruvate dehydrogenase. So if you have a coenzyme A and you have a pyruvate dehydrogenase, you can change a molecule of pyruvate into acetyl CoA. Now, acetyl CoA is a smaller molecule. It's a 2-carbon molecule, whereas pyruvate is a 3-carbon molecule. And there, the reason for it going from 3 to 2 carbon is that 1 molecule of carbon dioxide leaving. So in goes our acetyl CoA. Now it's small enough, it's only 2 carbons, small enough to cross the mitochondrial membranes, because remember the glycolysis cycle producing pyruvate happened in the cytoplasm. Now it can go across the mitochondrial membranes into the mitochondria, and it can take part in the citric acid cycle, which occurs in the matrix of the mitochondria. So right up there for yourself, a little note, it's in the matrix of the mitochondria. So when acetyl CoA goes into the citric acid cycle and you have the enzyme citrate synthase present, you can produce a molecule of citrate. So let me put these intermediate names in a little block for you so they stand out a little bit more. We have acetyl CoA and it goes and forms citrate if you have citrate synthase present. And then what we're going to see at this point is that coenzyme A is going to leave. All right, coenzyme A exits over there. So that's our first reaction, citrate forming from an acetyl CoA molecule. Now there is one more thing here that is needed for this to actually happen. We have to put in a molecule of H2O, 1 water molecule in here to make this happen. So put in your water molecule right there. Okay, let's move on. What happens next to citrate is interesting. It's going to be changed into a molecule called isocitrate. Isocitrate. We do need an enzyme for that to happen. That enzyme's name is aconitase. So then you'll change citrate into isocitrate, and this is an isomerization reaction. All right, moving along from isocitrate, next we're going to form a molecule called alpha keto glutarate. Alpha keto glutarate. If we do have the enzyme isocitrate dehydrogenase available, this reaction will happen. So isocitrate dehydrogenase, if it's there, it can form a molecule of alpha keto glutarate. All right, another thing that needs to happen here is that you need some reducing power. So you need one molecule of NAD plus to come in here and provide that reducing power, and it leaves as a molecule of NADH. The other thing that's interesting is going from an isocitrate to an alpha keto glutarate will mean that a molecule of carbon dioxide will leave. So this is the first time here in the Krebs cycle that we see that a molecule of carbon dioxide is leaving just before the alpha keto glutarate forms. All right, next up, alpha keto glutarate will undergo a change and form succinyl CoA. Succinyl CoA is that name of that molecule. Again, over here, we're going to need some reducing power. So we need one molecule of NAD coming in, and it leaves as NADH. Also happening here, another molecule of carbon dioxide is leaving. The enzyme that we need to make this happen is alpha keto glutarate dehydrogenase. Alpha keto glutarate, after the name of the intermediate, and it is a dehydrogenase, right? Because you have NADH playing a role here. Dehydrogenase. So you see back to back, these two reactions are dehydrogenase reactions. So here we have an oxidation, and here we have another oxidation to form succinyl CoA. All right, now that we have succinyl CoA, we'll see something interesting happen. You'll see a coenzyme came in again, right, to form that succinyl CoA. So a coenzyme comes in to form succinyl CoA, but then it's also going to leave again when the next molecule forms, which is called succinate. Succinate. All right. So if succinyl CoA, if it loses the coenzyme, it becomes succinate. It does need an enzyme here to help with that, and that enzyme's name is succinyl CoA synthetase. Succinyl CoA synthetase. All right. For the first time here, we're going to see the formation of a molecule of ATP. So you can draw it here on the inside or the outside. I'm just going to put it where I have space. I'm going to put it in red so it stands out. An ADP molecule is going to pick up a phosphate and produce an ATP molecule. You'll see in some textbooks they actually link this to a GDP forming a GTP and then an ATP. So it's not incorrect if you do that, if you draw in the arrow like this, GDP, GTP, and then it's going to form ADP and ATP, right? That's fine. If you just do the ADP, ATP, that's actually sufficient. I wouldn't mark off any points if you don't have this part in down here. All right, so from succinate, next we're going to move to a molecule of fumarate. So that's the name of the next molecule, fumarate. And we do need a dehydrogenase enzyme here as well. Succinate dehydrogenase is that enzyme's name. So that tells us that we need some reducing power, but this time it's not going to be NAD+, rather it is going to be FAD. FAD that comes in and it's going to pick up 2 hydrogens and it's going to form FADH2. Then that fumarate molecule will form. So again, this is an oxidation occurring over here. Succinate is oxidized into fumarate, but the FAD is reduced, right, into FADH2. Next up, fumarate is going to form a molecule called malate. Malate. If you have the enzyme fumarase present, that will happen. You also need to add a molecule of water over here. And this is the only place here and over here at the start where you're going to see a water molecule needed, right? Only those 2 places. So there you have malate. Fumarate was oxidized into malate. Next up, malate is going to form a molecule of oxaloacetate. Oxaloacetate. Now this is also an oxidation. So you're going to need some reducing power here again. And again here it will be NAD+ coming in, picking up a hydrogen there and oxidizing malate to form oxaloacetate. It can't happen without an enzyme. So put in the name of the enzyme. It's malate dehydrogenase. All right. And then this is going to complete our cycle, because what will happen next is oxaloacetate will form a molecule of citrate. So let's, add these arrows together here, up here at the top, like that. If you combine it with a molecule of acetyl Coa. Now take note here, this molecule, oxaloacetate, is a 4-carbon molecule. Citrate is a 6-carbon molecule. But how did we get to 6 carbon? Remember, if you take 4 carbons from oxaloacetate, add it to 2 carbons from acetyl Coa, you form a 6-carbon molecule. Right, remember we did have to add water here to make this happen as well. There is another thing I want to point out on the cycle for you. In a moment we'll talk about the energy in the cycle, but look carefully. Wherever you see NAD+ or an FAD come in and play a role to add some reducing power, your enzyme name ends with dehydrogenase. Right, so this is not too hard to learn. There's an isocitrate dehydrogenase, an alpha-ketoglutarate dehydrogenase, then a succinate dehydrogenase, and a malate dehydrogenase. So then the other enzyme names are easy to fill in. They're not going to be the dehydrogenases. All right, I said I want to show you something about the energy in this cycle. This is very interesting. I'm going to use a different color, and I'm going to use some abbreviations here. I'm going to use NE, and NE stands for near equilibrium. Let's use a different color that you can see better. So this is near equilibrium. And then there's going to be some endergonic reactions. Let's call them EN. And there's going to be some exergonic reactions. Let's call them EX reactions. So where do they fit here in on the cycle? Well, let me show you. Here, the first reaction going from acetyl CoA to citrate is a very, very exergonic reaction. I think I may have space to write it in. Very exergonic, right? So that's EX over there. Then going from citrate to isocitrate is a near equilibrium reaction. Moving between isocitrate and alpha-ketoglutarate, here again, we have an exergonic reaction, so EX. Between alpha-ketoglutarate and succinyl-CoA, again, we have an exergonic reaction. between succinyl and succinate, succinyl CoA and succinate, remember that's just simply a coenzyme leaving. So the energy stays basically the same. We don't have to fill in anything there, but to form our succinate, you know, is really a near equilibrium reaction. So you can put in Ne like that, that's fine. Moving between succinate and fumarate also is a near equilibrium. Moving between fumarate and malate, also near equilibrium. And then when we get here to moving between malate and oxaloacetate, you'll see this is the first endergonic reaction, En. So what does that tell us? It tells us that at the start here, where you have a very exergonic reaction, this in fact will drive the endergonic reaction forward. Because you know, an endergonic reaction means you have to put in energy, right? But if you release a lot of energy here on this side of the cycle, here where we had very exergonic and here, the energy released here can be used in this endergonic reaction here to push this one forward to form citrate again. So it's like a wheel that spins really fast here at the start, and then it spins so fast that it pushes the momentum over here so that the oxaloacetate will form a citrate molecule. All right, now you'll see in the textbook, if you look at your cell biology textbook, that they include usually the structure of pyruvate, the structure of acetyl CoA, and all of these intermediates you see here in these blocks. I will give you a bonus point for this cycle if you draw in the structure of these intermediate molecules, but it isn't required. I'm not going to deduct any points if you don't draw any of those structures in there. I do want to point out one last thing before we end. I said that this enzyme name down here is succinated, rather this one down here, succinyl CoA synthetase. Some textbooks will call this enzyme succinate thiokinase. Right, so if you want to remember that one, that's fine. I mean, I'll go with either or. Let's check if we have everything on this cycle. I believe we do. We have our 2 carbon dioxides leaving. We have our ATP molecule form. Yes, we have our 2 water molecules, and we have our 3 NAD pluses and our FAD. Remember, there was a carbon dioxide up there and an NAD plus here as well. This part of the reaction up here is known as the prep reaction. The prep reaction. Or sometimes they'll call it the prep step. There is also some textbooks that will call it pyruvate oxidation. So pyruvate oxidation has to happen before the Krebs cycle can occur down here. Remember, when we start with two pyruvates down here, all of this is going to happen twice. The cycle, the Krebs cycle, is going to spin twice. So put one nice big times 2 down here below your cycle, right? Because you're going to form 2 molecules of citrate from every pyruvate molecule that goes in.